Hello, good afternoon, viewers at home. My name is Olatunde Michael. I am the team leader of Omega Bio-Edutainment. This is another part of the educational series of our platform, and today we are going to look at a focus of microbial genetics. For the rest of the course, we are going to have the following topic, at least that we'll be treating in the next few videos. Like 8 or 10 videos will be dedicated for microbial genetics. And if you remember that the last video, I think sometimes last week, we started with understanding gene expression and gene regulation. After that, we will examine what recombinant DNA technology is, what are the basic mechanisms of recombinant DNA technology, or what we can also regard as genetic engineering. We are going to look at this. Then later on, we will proceed into understanding what DNA or RNA hybridization technique is, and we are going to look at the practical application of this technique to understanding what microbial or molecular genetics is all about. After that, there will be a dedicated video for microarray technology with short form maths. So we are going to look at this basically. We are going to look at the basic mechanism and its application of math technology as we have it. Then later on, we are going to look at polymerase chain reactions, PCR techniques. We look at the basic principles of PCR, the types of PCR, Sanger principles, and post-Sanger era. We are going to look at that. Then immediately after that, we will proceed into understanding sequencing techniques and its applicability in microbial genetics. We will look at the types of sequencing techniques. We will look at whole genome sequencing as well, and we'll look at its application. Thereafter, we will proceed into understanding genomics, bioinformatics principles, proteomics, and the world of omics and its applicability as a whole. So, thereafter, we will now look at some other basic techniques like ELISA technique, that is enzyme link immunosorbent assay. We look at spectrophotometry. We are going to look at that, and thereafter we will have to start picking them one after the other, and we are going to widen the scope as we have the advanced techniques of this. These are basic principles, laboratory techniques in molecular biology. And if you are a microbiologist today, you need to actually get yourself acquainted with this, because this is going to be part of what you will need to harm yourself in the world of microbiology today. And in diagnostic medicine or diagnostic microbiology in particular, all these principles are the basic that we actually make use of. And just like I said, in the next few hours, we are going to continue with this. Just to let you know the synopsis as we have it highlighted on the screen for you. I want to beg your attentive attention and make sure that you prepare very well so as to be able to go through all this. We are going to go through this throughout the festive period, and by the grace of God, by the time we are entering into 2025, I'm very sure that we'll be better off in terms of our understanding of basic molecular biology techniques. Once again, I remain the team leader of Omega Bio-Edutainment, and I want you to continue to like our videos. We also have others videos that we treat a lot of biological courses ranging from virology, bacteriology, immunology, genetics, and what have you. And I'm very sure that by the time you subscribe, we are going to have a lot of time together. And just like I said, certain period will be dedicated for tutorials online, and we'll begin to treat some basic past questions. At least in each of these courses, we are going to have nothing less than 50 questions that we will be